This is an overview of an article entitled Variants in NKX2-5 and FLNC Cause Dilated Cardiomyopathy and Sudden Cardiac Death, published in the August 2018 issue of Circulation, Genomic and Precision Medicine. Dilated cardiomyopathy, or DCM, is a morphological classification of heart muscle disease exemplified by progressive ventricular dilatation, impaired function, heart failure syndrome, and a risk of sudden cardiac death. In fact, sudden cardiac death can be the sentinel event, making diagnosis of DCM arduous unless a comprehensive post-mortem necropsy is performed. Approximately one-third of cases have a positive family history for DCM, and in the majority of cases, the mode of inheritance is consistent with an autosomal dominant disorder with age-dependent penetrance and variable expressivity. Where genetic testing is performed, a putative pathogenic variant is only identified in 50% of cases, with the majority being genetically elusive. Variants in over 50 genes have been associated with DCM and include genes encoding cytoskeletal, sarcomere, Z-band, nuclear membrane, and intercalated disc proteins. Of all the monogenic cardiovascular diseases, the genetics of DCM is arguably the most complex, with DCM phenotypes seen in cases with genetic variants associated with other cardiomyopathies, ion channelopathies, muscular dystrophies, and mitochondrial disorders. It's increasingly recognized that conduction system disease, bradyarrhythmias, and tachyarrhythmias can precede overt structural changes to the heart, highlighting the limited understanding of phenotypic expression and genetic pleiotropy. The Icelandic population comprises a unique study cohort which is geographically isolated, with well-maintained census and electronic medical records data, lending itself to genetic epidemiological studies, including the ability to determine founder effects. The authors of the paper under discussion searched for variants causing DCM in Iceland and reported on genotype-phenotype associations. They performed a genetic case control study with 424 cases of DCM and 337,689 population controls. They used a combination of whole genome sequencing, single nucleotide polymorphism chip genotyping, and imputation. They then performed logistic regression analyses to test 32.5 million sequence variants for association with DCM or secondary traits, such as electrocardiographic abnormalities, which can precede left ventricular dilatation and dysfunction. Genome-wide significant associations were identified for two rare variants. The first was a missense variant, phenylalanine 145 leucine, in the NKX2-5 gene with an allele frequency of 1 in 7,100 Icelanders. NKX2-5 is a homeobox gene that encodes a transcription factor important in cardiac development and is associated with congenital heart defects, most frequently atrial septal defects, conduction system disease, severe enough to warrant a pacemaker, ventricular arrhythmias, and more recently, DCM. The second identified variant was a frameshift variant in the FLNC gene with an allele frequency of 1 in 3,600 Icelanders. FLNC encodes filamin C, a large actin cross-linking protein that's expressed in both cardiac and skeletal muscle, anchors protein complexes at the sarcolemma, Z-discs, and intercalated discs in cardiomyocytes, and provides a scaffold for cytoplasmic signaling proteins. FLNC has recently been associated with DCM and neuromuscular disorders, in particular myofibrillar myopathy. Here's a table with phenotype data of the participants with the NKX2-5 and FLNC variants. Both were associated with DCM and sudden cardiac death. However, NKX2-5 carriers were more likely to have congenital heart defects and advanced conduction system disease and receive a pacemaker. FLNC carriers had a higher frequency of skeletal myopathy. In this 6-generational pedigree, 23 individuals carried the NKX2-5 variant and share an ancestor born in 1865. Those expressing the phenotype of DCM and/or atrioventricular block are colored blue. 13 patients had medical records, of whom 3 were deceased. 8 had cardiomyopathy. Of note, all 8 patients with cardiomyopathy also had conduction disease, with 5 diagnosed with conduction disease prior to a diagnosis of cardiomyopathy. 6 patients had pacemakers, and 3 had implantable cardioverter defibrillators. 3 genotype-positive individuals did not express DCM or conduction system disease at this time. Similarly, this pedigree of FLNC variant carriers traces back to a common ancestor born in 1595. 50 carriers of this variant were identified, of whom 38 had medical records available. Of these, 9 had cardiomyopathy. 3 carriers had sudden cardiac death events, and 12 carriers had conduction system disease and/or arrhythmias. Finally, the authors assessed for the association of electrocardiographic abnormalities in either the NKX2-5 or FLNC variant carriers. As shown in this heat map, the most notable abnormalities were decreased R-wave amplitude, PR interval prolongation, and T-wave amplitude. Other known NKX2-5 or FLNC variants did not have strong effects on these traits. In conclusion, the authors identified 2 rare novel variants in NKX2-5 and FLNC that cause adult-onset DCM and are associated with sudden cardiac death in the Icelandic population. Taking the 2 variants together, 1 in 2,400 Icelanders carries either of these mutations. Both variants are associated with cardiac conduction system disease and arrhythmia. However, NKX2-5 expresses a more severe phenotype and is more likely to show more severe conduction system disease in the form of high-grade atrioventricular block, as well as atrial septal defects. Given that conduction system disease can precede structural heart disease, particularly for NKX2-5 carriers, genetic and clinical screening may lead to timely intervention and the prevention of sudden cardiac death.